And I think we will start with a presentation by Dr. Eva Gelinsky, who is joining us online. Dr. Gelinsky is an independent scientist from Switzerland. She's an expert on seed policies and science. She's also working as policy coordinator of the Initiative for GMO-Free Seeds and Breeding. She is a member of a federal ethics committee on non-human biotechnology in Switzerland, and with her presentation, she will dissect precisely this narrative on sustainability of novel genomic techniques, such as drought resistance. Good morning, Eva. Are you with us?
Good morning, everybody. Thank you very much for the introduction. Well, I don't know if is my presentation. Okay, it's coming. Perfect. Yes, as we already heard, new genetic engineering methods are being brought into play as a solution in order to meet the challenges of climate change. Next slide, please. The new genetic engineering methods, such as CRISPR, enable, among other things, several simultaneous changes within the genetic material, which are hardly possible with conventional methods. This is linked to the hope of accelerating research and breeding in order to find advantageous characteristics and develop new combinations more quickly than before. The techniques are used, for example, to study the regulation of genes and their involvement in the response to climatic stress factors. However, generating stress tolerance in plants by means of genetic engineering will remain a challenge, even with the new methods. For there is no single trait, for example, drought tolerance, that can be bred for or even integrated into plants. To stay with the example of drought tolerance, plants have numerous ways of reacting to a lack of water. They can, for example, root deeper, root wider, grow more fine roots, grow a thicker wax layer on the leaves, or more leaf hair to reduce evaporation. Which of these reactions plants are capable of, or particularly capable of, depends within their species characteristics on the genetics of the individual plant or variety. Each of the characteristics just mentioned, for example, the ability to root deeper, does not in itself make a drought-tolerant plant. From this, it follows first of all, breeding for drought tolerance is not a simple matter, but a highly complex one, because the stress response of plants is multidimensional, involving various signaling pathways regulated, for example, by phytohormones and other volatile organic components. Next slide, please. This network must first be understood in detail, but also precisely in its complexity. Next slide, please. Genome editing methods have so far been used mainly in basic research to study the complex interplay of different signaling pathways. For this purpose, mainly small changes are made to the genome. Stress genes are switched on or off, for example, in order to better understand what role they play in the plant's stress response. Next slide, please. This leads to individual findings, but not to an understanding of the complexity as a whole. Furthermore, an intended intervention in the complex interplay of different signaling pathways within the cell with genome editing techniques can have an impact on many other properties. Second, traits that contribute to a certain drought tolerance are anchored in the overall constitution of plants. This means that improving drought tolerance through breeding is almost always linked to further fundamental plant physiological changes. Deeper root growth, for example, consumes energy that the plant actually needs elsewhere to grow. This is why, for example, crop yields are reduced, growth is slowed down early, quality suffers, and the grains are smaller. That means if we want to breed drought-tolerant plants, we must therefore be prepared to adapt to other types of plants and change our cultivation and food habits accordingly. It will not work to think that the type of high-yielding variety that dominates agriculture today can simply be equipped with additional drought tolerance. But there's another problem. In Europe, too, we are increasingly confronted with strongly fluctuating climatic conditions. Even if it currently looks like the spring and summer droughts will increase, there may still be some very wet years in the future. In addition, there will be an increase in extreme events such as heavy rainfall or very high temperature. The problems triggered by these fluctuations cannot simply be solved by equipping plants with drought resistance or specific disease resistance, regardless of the breeding method. The volatility of climatic conditions is decisive. It will hardly be possible to genetically equip a single variety in such a way that it can produce maximum yields in all expected climatic extremes. In addition, at the beginning of the year, when the farmer has to decide on a variety, he does not know what the growing season will be like. Dry, wet, very hot, or rather cool? Next slide, please. The good news is that there are already solutions available that could be implemented very quickly. I will just give one example. It is possible to grow different varieties of a species, such as wheat, in a mixture. The yields and quality characteristics of these mixtures are often better than the average of their pure seeds. This means that by cultivating in mixtures, more can be harvested than if the respective varieties were cultivated separately. The different varieties have different characteristics. Varieties with long roots absorb water from deeper layers in dry conditions. Varieties with shallow roots absorb surface water better in wetter years. If the varieties are grown together, the mixture has both properties and is more stable in yield. The differences between the mixture partners can increase overall resilience, that, for example, the ability to withstand weather extremes. Next slide, please. I will sum up. In view of the effects of climate change that can already be observed and the developments that are emerging globally, there is a great urgency to adapt agriculture in Europe to the rapidly changing conditions. Due to the complexity of climate-relevant traits I just described, it cannot be assumed that the new genetic engineering approaches can contribute to the adaptation of agriculture to global warming. It is not to be expected that climate-adapted varieties developed using CRISPR-Cas will be available quickly, if at all. Next slide, please. This is also emphasized by the Swiss Ethics Committee on Non-Human Biotechnology in its latest report. The clear majority of members are skeptical about the ability of new genetic engineering approaches to make a relevant contribution to the adaptation of agriculture in the required timeframe. Next slide, please. On the other hand, various solutions are already available to adapt agriculture to the rapidly changing conditions. Next slide. But in this context, it is necessary to avoid path dependency in both research and practice. Other research and breeding approaches must be neither neglected nor impeded by investing in only one technological approach. For precautionary reasons, they must be organized in such a way that multiple paths remain open for agriculture to fulfill its ethically indispensable tasks, namely ensuring adequate food as well as protecting biodiversity. Thank you for your attention.
Thank you very much, Dr. Gelinsky. And indeed, I think any organic farmer would be able to tell you that to have a resilient farming system, first of all, you would need a healthy soil. And the whole idea that you could claim that a product is sustainable on the basis of a trait or a given variety is certainly a bit simplistic. So thank you for clarifying these aspects.